Tuesday, July 22nd, 2025, AGNDD intelliReport topic. Hippocampus. Please pardon any visual and informational inaccuracies in this entirely AI-generated Wikipedia-sourced video article. Explore the fascinating history and evolving understanding of the hippocampus, a crucial brain structure linked to memory and emotion, from its early descriptions to modern insights. The hippocampus, a brain structure, was first described by Julius Caesar Aranzi in 1587, who compared it to a silkworm and a seahorse. The term hippocampus comes from the Greek for seahorse. Over the years, various anatomists proposed different names, such as ram's horn and cornu ammonis. The hippocampus was associated with memory in the mid-20th century. A naming error involving hippopotamus was corrected by Carl Friedrich Burdach in 1829. The term hippocampus minor was removed from anatomical texts in 1895, and today the structure is known simply as the hippocampus, with subfields named CA1, CA4. The hippocampus, part of the limbic lobe described by Broca in 1878, is a key component of the limbic system, which includes the cingulate and parahippocampal gyri. Initially linked to olfaction, later studies by Papez and MacLean highlighted its role in emotion. The hippocampus connects to brain regions involved in emotional behavior, such as the septal area, hypothalamic mammillary bodies, and the anterior nuclear complex in the thalamus. MacLean suggested these interconnected structures form the limbic system, emphasizing their collective involvement in emotional processes. The hippocampus is a three-layered archicortex structure within the medial temporal lobe of the brain, involved in spatial memory and navigation. It stretches from the amygdala to the splenium of the corpus callosum and includes subdivisions like the dentate gyrus and subiculum. The hippocampal formation comprises the hippocampus and related structures like the entorhinal cortex. Despite its similar appearance across mammals, its size relative to body size varies, being larger in primates compared to rodents. The hippocampus's volume correlates with spatial memory capacity, with species having better spatial memory often possessing larger hippocampal volumes. The hippocampus, a key brain structure, has a curved, rolled-up tube shape and includes subfield CA1, CA4, with CA4 sometimes describing part of CA3. It features densely packed pyramidal neurons forming a U-shape with the dentate gyrus embedded in it. In humans, the hippocampus has anterior and posterior parts, while in primates and rodents these are termed differently. The hippocampus and dentate gyrus form a banana-like structure connected by the commissure of fornix. The entorhinal cortex, EC, links the hippocampus with cortical and subcortical structures, playing a crucial role in memory. Complete amnesia occurs if both the hippocampus and parahippocampus are damaged. The hippocampus receives major input from the entorhinal cortex, EC, and sends output via CA1 to the subiculum. Information reaches CA1 through direct and indirect pathways. The direct pathway involves axons from EC layer 3 forming synapses on CA1 dendrites, while the indirect pathway involves a trisynaptic circuit through the dentate gyrus and CA3. Basket cells in CA3 provide inhibitory feedback to pyramidal cells, moderating excitatory responses. Additional outputs connect to cortical areas and the hypothalamus. The hippocampus receives modulatory inputs from neurotransmitter systems and the medial septal nucleus, crucial for hippocampal function and memory processing. The hippocampus is divided into subfields and subregions, head, body, tail, with distinct anatomical and functional roles, connecting differently to other brain regions. In humans, these are termed anterior, intermediate, and posterior hippocampus, respectively, with the posterior region involved in spatial and verbal memory. The dorsal hippocampus in rats, rich in place cells, is crucial for spatial memory. Historically, the hippocampus was thought to be linked to olfaction, but later studies showed its involvement in memory for odors. The intermediate hippocampus shares traits with other regions, while the ventral hippocampus is associated with fear conditioning and affective processes. Theories on hippocampal function include response inhibition, episodic memory, and spatial cognition. Initially, response inhibition suggested the hippocampus controlled hyperactivity and response inhibition, evolving into Jeffrey Gray's behavioral inhibition system theory. The memory theory gained prominence after the case of Henry Molaison, who suffered severe amnesia post-hippocampal surgery. The spatial cognition theory, introduced by E.C. Tolman and expanded by John O'Keefe, identified the hippocampus's role in spatial memory through place cells. Efforts to reconcile these theories suggest the hippocampus integrates memory and spatial functions, supporting cognitive mapping and navigation, with neuroimaging studies exploring its role in approach avoidance conflict. The hippocampus is crucial for forming explicit memory, including episodic and semantic memory, and encodes emotional context from the amygdala. It exists in both cerebral hemispheres, and damage to both can cause severe memory issues like anterograde and retrograde amnesia. Memory consolidation may involve transferring memories out of the hippocampus. The hippocampus is essential for forming and recalling, but not storing memories. It doesn't affect procedural memory, which relies on different brain regions. The hippocampus's role in implicit memory is debated. In rats, intense learning events cause DNA methylation changes in the hippocampus, with long-term memory stored in the anterior cingulate cortex. The between systems memory interference model suggests that the hippocampus inhibits non-hippocampal memory systems during its activity. When inactive, non-hippocampal systems consolidate memory, but these memories aren't recalled upon hippocampal reactivation. This model highlights the hippocampus's dominant role over other memory networks, especially with incongruent information. It suggests future research could focus on non-hippocampal systems via hippocampal inactivation, expanding understanding of memory. The model also contributes to theories like the multiple trace theory, offering a broader perspective on memory processes, indicating that memory effects may involve multiple brain regions, not just the hippocampus. The brain's navigational cells, primarily in the hippocampus or connected to it, include place cells, speed cells, head direction cells, grid cells, and boundary cells. These cells form a network crucial for spatial memory. Place cells, discovered in the 1970s, support the idea of the hippocampus as a cognitive map. Dysfunctional hippocampus affects orientation, leading to disorientation and memory issues. Studies on animals and humans show that place cells are essential for spatial tasks and navigation. They interact with other cells to provide spatial information, and their activity is linked to theta waves and various task-related variables. Approach avoidance conflict involves decision-making in situations that can be both rewarding and punishing, often linked to anxiety. fMRI studies reveal that the anterior hippocampus is sensitive to conflict, playing a role beyond long-term memory or spatial cognition. It is part of a broader network crucial for decision-making under uncertainty. A review highlights multiple studies showing the hippocampus's involvement in conflict tasks. The challenge is to understand how conflict processing relates to spatial navigation and memory, suggesting these functions may not be mutually exclusive. Recent studies highlight the hippocampus's crucial role in social memory. In epileptic patients, specific hippocampal regions show unique neuronal responses to familiar faces. Rhesus monkeys also exhibit hippocampal mapping for facial and vocal identities. MRI studies reveal that CA1 and CA3 neurons respond to social stimuli, while CA2's role remains less clear. However, genetic inactivation of CA2 neurons in mice results in significant social memory loss without affecting sociability. Similarly, ventral CA1 neurons are essential for social memory, as demonstrated through optogenetic experiments. These findings underscore the hippocampus's involvement in processing social memories. The hippocampus exhibits two main activity modes, theta and large irregular activity, LIA. Theta mode, characterized by large regular EEG waves, 6 to 9 Hz, occurs during active behavior and REM sleep, with sparse neuron activity. LIA mode, featuring sharp EEG waves, appears during slow-wave sleep and waking immobility, with increased neuron firing during sharp waves. In rats, theta mode involves spatially influenced cell activity, while LIA shows correlated neuron firing with sharp waves. These modes are also present in primates, although robust theta rhythmicity is less evident, but similar sharp waves and neural activity changes are observed. Theta waves, generated by neural layers in the entorhinal cortex and hippocampus, are prominent EEG signals known as hippocampal theta rhythms. These waves, occurring at 3 to 10 Hz, modulate hippocampal neuron activity and synchronize across the hippocampus. The trisynaptic circuit in the hippocampus is thought to generate these rhythms, which are observed in both rodents and humans. In rats, theta waves appear during active interaction with the environment and REM sleep. While their function is unclear, they are hypothesized to relate to learning and memory through synaptic plasticity. Lesions in the medial septum disrupt memory, indicating its role in theta rhythm regulation. During sleep or rest, the hippocampus exhibits irregular slow waves and sharp waves, which are linked to bursts of activity and high-frequency oscillations called ripples. These sharp waves, more frequent during sleep, are associated with memory reactivation, where neural patterns memorized during behavior are replayed. This process supports the two-stage memory theory, suggesting memories are stored in the hippocampus during activity and transferred to the neocortex during sleep. Sharp waves facilitate synaptic changes crucial for memory consolidation, though their suppression can affect memory expression. Despite this, new place cell codes can still form in non-demanding tasks. Long-term potentiation, LTP, is a key mechanism believed to underlie memory storage in the brain, first speculated by Ramon Y. Cajal and formalized by Donald Hebb. In 1973, Tim Bliss and Terje Lomo observed LTP in the rabbit hippocampus, characterized by sustained synaptic responsiveness following strong activation. LTP, heavily studied in the hippocampus, particularly in the CA1 region, involves glutamate and NMDA receptors, which allow calcium entry during simultaneous pre- and postsynaptic activity. Blocking NMDA receptors or disabling LTP in genetically modified mice leads to memory deficits, highlighting LTP's crucial role in memory, especially spatial memory. Research on a hippocampal prosthesis brain implant has been ongoing since the early 2000s. By 2018, a nonlinear multi-input, multi-output model, MIMO, was developed, showing promise in restoring and improving memory function. A modified version, the memory decoding model, MDM, has demonstrated potential for significant memory modification. Future research is suggested to evaluate both models, with a focus on the hippocampal theta wave input. Normal aging often leads to declines in episodic and working memory, potentially linked to changes in the hippocampus. While early studies suggested significant neuron loss in the hippocampus of the elderly, later research found minimal differences. MRI studies have shown mixed results regarding hippocampal shrinkage with age. However, there is a consistent correlation between hippocampal size and memory performance, with shrinkage leading to impaired memory. Additionally, elderly individuals show less hippocampal activation during memory tasks compared to younger people. A 2011 study revealed that aerobic exercise can increase hippocampal size and enhance spatial memory in older adults. In Alzheimer's and other dementias, the hippocampus is one of the first brain regions affected, leading to symptoms like short-term memory loss and disorientation. Amyloid beta deposits initially appear in the frontal lobes, followed by neurofibrillary tangles in the hippocampus. Damage to the hippocampus can also occur due to hypoxia, encephalitis, or medial temporal lobe epilepsy, potentially causing anterograde amnesia. Dementia often results from cerebral ischemia, which alters the hippocampus, particularly the CA1 area responsible for episodic memory. This change leads to episodic memory impairment, an early symptom of post-ischemic dementia. The hippocampus is highly susceptible to long-term stress due to its abundance of glucocorticoid receptors. Chronic stress, elevated cortisol levels, and conditions like Cushing's syndrome lead to hippocampal atrophy, affecting disorders such as PTSD, schizophrenia, and depression. Stress-related atrophy can be mitigated by antidepressants and cortisol-reducing treatments which promote dendritic restoration. Socioeconomic factors like family income influence hippocampal volume in children through stress. Sex-specific responses to stress show males experiencing more hippocampal damage than females, likely due to protective ovarian hormones. Early life stress can have lifelong impacts on hippocampal function, with evidence primarily from rat studies. Research indicates a link between reduced hippocampus volume and PTSD. Vietnam War veterans with PTSD showed a 20% reduction in hippocampus size compared to those without PTSD. However, this was not observed in individuals with chronic PTSD from a 1988 airshow crash. Interestingly, non-combat twin brothers of Vietnam veterans with PTSD also had smaller hippocampi, suggesting a potential genetic or pre-existing factor. A 2016 study supports the idea that a smaller hippocampus may increase PTSD risk, while a larger hippocampus could enhance treatment effectiveness. This highlights the complex nature of the relationship between hippocampus size and PTSD. Transient global amnesia, TGA, is characterized by sudden temporary anterograde amnesia and variable retrograde memory loss, typically lasting up to 24 hours. Diagnosis is confirmed through DWI MRI, which reveals small lesions in the CA1 subfield of the hippocampus, detectable 24 to 96 hours post-onset and resolving within 6 months. These lesions, 1 to 5 mm in size, may be single or multiple and affect either hemisphere, impacting verbal or visuospatial memory depending on the side. The selective vulnerability of CA1 neurons suggests metabolic stress as a potential cause, with other theories including venous reflex, ischemia, epilepsy, migraine, or network connectivity issues. The hippocampus is a brain region where new neurons are generated, specifically in the dentate gyrus. Neurogenesis can be enhanced by exercise but hindered by epileptic seizures. Temporal lobe epilepsy may disrupt neuron development and cause tissue damage, with hippocampal sclerosis being common. It's unclear if epilepsy results from hippocampal abnormalities or if seizures damage the hippocampus. Experiments show that induced seizures often lead to hippocampal damage, possibly due to excitable glutamate receptors causing hyperexcitability, cytotoxicity, and cell death. This damage might also relate to the hippocampus's ongoing neurogenesis and potential abnormalities in this process. Hippocampal volume reduction is a consistent finding in schizophrenia, particularly affecting the left side and evident from the prodromal stage. This reduction, independent of antipsychotic treatment, is linked to psychotic symptoms, possibly due to altered dopamine release affecting the prefrontal cortex. The CA3 and CA2 subfields show the greatest volume reduction, correlating with memory dysfunction and decreased connectivity with the prefrontal cortex. Dysfunctional GABA transmission and reduced M4 muscarinic receptors are noted, with reduced neurogenesis in the dentate gyrus. Hyperactivity, especially in the anterior hippocampus, is linked to psychosis with molecular changes in CA1 and CA3 subfields. Hippocampus atrophy is observed in individuals with microcephaly. Mouse models with WDR62 mutations, mimicking human point mutations, demonstrate impaired hippocampal development and neurogenesis. Non-mammalian vertebrates lack a hippocampus-like structure visually, but possess a homologous one, part of the allocortex. The mammalian hippocampus evolved from the pallium, present in all vertebrates, including primitive ones like lampreys and hagfish. The pallium divides into medial, lateral, and dorsal zones, with the medial pallium as the hippocampus precursor. Despite lacking the distinctive S-shape and enfolding of the dentate gyrus, strong chemical and functional similarities indicate homology. Evidence suggests these hippocampal-like structures play roles in spatial cognition in reptiles and fish. In teleost fish, the forebrain is everted, with structures on the outside, unlike other vertebrates. This results in the medial pallium, the hippocampal zone in typical vertebrates, corresponding to the lateral pallium in fish. Goldfish, in particular, exhibit strong spatial memory and can form cognitive maps. Damage to both the lateral and medial pallium impairs their spatial memory. It's unclear if the medial pallium has a similar function in basal vertebrates like sharks and rays. The dorsolateral pallium in teleosts is homologous to the hippocampus in terrestrial vertebrates. In 2023, goldfish brain mapping showed telencephalon subregions akin to mouse hippocampal subfields. Birds, especially those that store food, have strong spatial skills linked to a brain region called the avian hippocampus. This area is similar to the hippocampus in other animals and is larger in birds that cache food. Damage to this region impairs their spatial memory. Insects like cockroaches and mollusks, such as octopuses, possess strong spatial learning and navigation abilities distinct from mammals, indicating no common evolutionary origin. In insects, mushroom bodies are linked to learning and memory, akin to the mammalian hippocampus. The octopus brain, organized in a circular lobe arrangement around the esophagus, features a vertical lobe involved in long-term memory. This lobe is analogous to the mammalian hippocampus and cerebellum and shares functional similarities with insect mushroom bodies. In summary, the hippocampus has evolved in our understanding from its anatomical roots to its crucial role in memory and emotion, forming a vital part of the limbic system.
Thank you for watching this AGNDD intelliReport, dear viewers. Please like, subscribe, and click the bell button to get notified during new uploads. And make sure to tune in next time. Stay safe and healthy. Cheers!